Good afternoon. Welcome to our presentation. We had about the water quality of Kyrkjetangen in Nordåsvannet. Our group members are Shishi, Marte, Anders, Wenche, Trude, and myself, Tom. Introduction. The study was a collaborative effort between the University of Bergen and Bergen municipality. It's important to monitor water quality to ensure public health at Kyrkjetangen and also to make sure that it's safe for swimming. We had two goals of our study. The first goal was of course to analyze the water quality of Kyrkjetangen, and the second goal was to make a recommendation for how water quality should be monitored in the future. We measured a lot of different water quality parameters, TCB, thermo-tolerant coliform bacteria, which in high concentrations could cause disease, biodiversity, phosphate, and also some physical properties, among which pH is important. The picture here says that the creek is polluted and that they advise not to swim here. That's from the Department of Health. Materials and methods. Here you can see our study site, and here is downtown Bergen, and here is Kyrkjetangen to the south of Bergen. So our sampling design. We took samples from 9 different locations, and here is a beach, and here is the second beach. And at one, here was a sign that you saw in the beginning that warned people to swim. And also there was an outlet here. At 5 and 9, there is diving boards. So, sampling. We sample for all the parameters at all the sites, and at the 2 beaches. We sampled for biodiversity. And we took TCB at 1 meter and surface, and we took in plastic bottles, and also we took phosphate to the samples, plastic bottles, and also we took the sediment samples from the 2 beaches in using a shovel, and took them into a bucket, and the different physical properties, like we use different apparatuses for.
Result and discussion. Here is our friendly E. coli, Mr. E. coli. For TCB, we make a plot about the surface water sample and 1 meter water sample. From the plot, we can know there is a green line, and the E. coli number for half of the water sample are below 100. It shows the water is in good quality. But the rest of the water sample, the TCB number are between 100 and 1000. So the water is in sufficient quality. The results are obtained according to European standard. For the 1 meter water sample, we can know that the TCB level in set 3 is obviously high, but quite low in set 7. As to the surface water sample, the TCB number is in set 6, 8, and 9 is higher than other sites. The probable reason may be that site 3, 6, and 8 belong to the beach, and there is a diving board in site 9. All of these sites are easily to be affected by human activities, so the pollution may be the reason for high level of TCB. We also make a box plot for the 1 meter water sample and the surface water sample as a whole. We find there is no significant difference between the water samples. So the reason may be due to the current and wind, so the water is well mixed. There is also no significant difference between different sites. It's probably because the sample size is too close.
Yeah, and we also decided to compare our data from our TCB data from 2012 against previous reports, but there was only one from 2006. So we compared it against that one. But also, since there was no significant difference between 1 meter and surface in TCB values, we took the mean of each site and compared it to 2006. And you can see at all sites, TCB values have gone up since 2009. So we thought maybe this had to do with rainfall. So we checked the rainfall on the 5 days before sampling for 2006 and 2012. You can actually see that there was slightly more rainfall in 2006, the days before sampling, so we can't really conclude with this. But as those of you who were in Bergen in 2012 in the summer could probably remember that it was raining a lot the entire summer. And through the set, it was sunny all summer of 2006. You could remember it because you had an outside summer job. We think maybe it has to do with the increase in rainfall during the longer time period of the entire summer of 2012. Or it might just be, since there is a general population increase in the area, that this has infected the TCB values.
Yes, and then over to phosphate and physical properties results. The different phosphate samples showed concentrations ranging from 0.015 to 0.026 milligrams per liter. Normally less than 0.020 milligrams per liter describes a water basin with very low phosphate concentrations. And our average concentration between the 9 different sites was 0.018, which means that phosphate concentrations at Kyrkjetangen are very low. We also measured the pH and found that at all 9 sites, the pH value was below 8. And we think there's a correlation between the low phosphate concentrations and the pH values being below 8, because phosphate naturally enters a water basin via sediment release through the chemical weathering of rocks, and pH affects the exchange of phosphate between sediment and water, and at values below 8, pH values below 8, which all of our values were, the phosphate bindings to metals in the rocks are stronger, causing less phosphate to be released from the sediment into the water.
Now we'll look at the biodiversity. This table shows the sediment samples that we found in at Kyrkjetangen. We found 5 different species, which polychaete was the one with the highest abundance found at site 3 and site 7. And here we can see a picture of the rest of the species, including polychaete. Yes. And then we used the Shannon index to calculate the H value, which gives us a clue about how the biodiversity in the area was. Since all the sites except for site 3 had only 1 species, the H value is calculated to 0. At site 3, the H value was 0.204, and then we also calculated total H value of all the species together, which was 0.302, which is a very low biodiversity. And then we compared site 3 and site 7 with the TCB results to see if there was any correlation. The TCB result at site 3 was very high, but at site 7 it was very low, so there wasn't any correlations there. And then we also looked at the phosphate results, which was also low, limiting the primary production, and this could also be a factor on the low biodiversity found at Kyrkjetangen. We also looked at the habitat, and Kyrkjetangen lies openly to the Nordåsvannet, which could cause a regular disturbancy to the benthic zone and make or cause a challenged environment for species living there, and also affect the low biodiversity.
And then a couple of recommendations, conclusions in the end. In general, the water quality at Kyrkjetangen is good. TCB values are good to sufficient, and phosphate concentrations are low, not any phosphate pollution or anything. But considering the fact that values have gone up from 2006 to 2012, we recommend that values are being monitored carefully in the future to prevent harmful conditions. And it would also be interesting to get future phosphate results so that we can, or so that they can be compared to the ones that we measured this year, because earlier Kyrkjetangen studies haven't focused on phosphate concentrations at all. And that way we can look at or see if the phosphate concentrations at Kyrkjetangen are stable, or if they fluctuate with time. And then we've asked ourselves, can the Kyrkjetangen area take more residents? At the moment, with these values, we would like to say yes. But again, considering the fact that TCB values have gone up the past years, or from 2006 to 2012, maybe one should be careful with building or constructing too much in the future. And at least the construction work should be coordinated with the monitoring of the water quality. Thank you. Any questions? Thank you to Christoph and Karen and everyone that helped us.
And thank you for making me feel safe to go swimming at Kyrkjetangen again.
Yep.
Okay, the next